everyone, welcome back to the cell signaling course. In our previous class, we saw how receptor serine threonine kinase signal in the animal world, and we draw a parallel how these receptor serine threonine kinases, which were called receptor-like kinases in plant kingdom, how they fared with respect to the signaling which is activated downstream. One key feature what we noticed was that the canonical pathway or the activation of receptor serine threonine kinase did not directly go through the MAP kinase pathway. We will see more details of receptor serine threonine kinase signaling in animal world, taking specific examples related to the TGF-beta signaling, bone morphogenetic protein signaling, activin signaling, and another class of receptors which respond to anti-Mullerian hormone. So, a small recollection of what we discussed as canonical serine threonine kinase receptor signaling. It is heteroterameric assembly where receptor type 2 has the kinase activity and the receptor type 1 has the site where phosphorylation can happen, and this heterotetrameric assembly is activated by binding of the ligand. And in canonical receptor serine threonine kinase signaling, what is first molecule engaged in perpetuation of the signaling is the Smad kind of proteins. There are multiple Smad kind of proteins. The ones which directly bind to this receptor are called R-Smad. And there is an interesting story of why these Smad molecules are called Smad, because it is a combination of two words, which one is a small and other one is mad. And if you recollect, in the plant receptor serine threonine kinase signaling, when we were discussing different receptor, I emphasized that how these receptors are named by the mutation phenotype. Similarly here, when people were studying different pathways, they found a set of mutants where organisms were small in size, or in different organism like Drosophila, people found that there are certain cells which are behaving in a way that they were named as mothers against daughters or decapentaplegic. So the combination of the two leads to this word which is called Smad. In that class of Smad, you have multiple different kind of molecules. The one which binds and associates with receptor is classified as R-Smad. This R-Smad is phosphorylated by the activity of the receptor serine threonine kinase, and which allows the dimerization of R-Smad, which requires another Smad molecule or co-Smad. Now together this complex of R-Smad and co-Smad, they move inside the nucleus where they regulate the transcription. We saw how CBP and P300 mediated signaling or the transcription was regulated. And when we say regulate transcription, it may activate certain genes, it may suppress certain genes. This is how the canonical signaling pathway moves on. Then what are the regulations which are present over there? In previous class, we saw that these Smad molecules, they have different key features which allow them to be regulated. One was that their presence inside the nucleus is mediated by the presence of the signal called NLS, and they are taken out of the nucleus once they have performed their job by the presence of the signal called nuclear export signal. In addition, their availability and binding to the receptor is regulated, their levels are regulated by different means. So in one scenario where this has to be regulated, the protein which is called SMURF and which is kind of E3 ubiquitin ligase, that works on the R-Smad and degrades it, as well as in combination with another class of Smad molecule which are called inhibitory Smad. Or I-Smad. R-Smad is not binding to the receptor because of the presence of these inhibitory Smad, as well as if their levels are down regulated by SMURF activity. There are multiple other signaling which contribute to this regulation on the canonical receptor serine threonine kinase signaling. Multiple signal which are through BMP or tumor necrosis factor or interferon or epidermal growth factor. They, along with this activation of this receptor serine threonine kinase, can activate the molecules like NF-kappa B, if you recollect we discussed that, or MAP kinases. This is something which we saw in case of plants getting activated in response to receptor serine threonine kinase activity. So these activities by different signals causing activation of these effector molecules are activating the iSmad. This iSmad ensures that rSmad is inhibited. Recollect in the previous slide we said that binding of rSmad to the receptor is regulated by either rSmad or by that class of molecule which was SMURF. So when you have canonical signaling going on, this inhibition is relieved, but normally you can have other signals which contribute to the activation of iSmad which inhibit this canonical receptor serine threonine kinase signaling. Then if you saw this molecule coming into picture through some of these ligands which are very specific to a receptor serine threonine kinase, are there non-canonical pathways of receptor serine threonine kinase which are activated? So what we know as a normal canonical pathway, I have dealt with this a couple of times now, but idea was that this complex of rSmad phosphorylated as a dimer and the SMAD4 or the co-SMAD, they go inside the nucleus and do the signaling. What happens in case of non-canonical RSTK signaling? Basically, you have multiple of these signals which interact with the receptor and other pathways and activate different set of molecule, like here it is a phosphatase PP2A, there is this RAS molecule, RAS is a small GTPase, we discussed this long time back when we were discussing G proteins and their importance in the signaling process as such. This RAS GTPase is known to work through a MAP kinase pathway and promote tumor-like growth. So what you notice is that in this case, the non-canonical signaling which is mediated by receptor serine threonine kinase is working through this classical MAP kinase signaling pathway as well. In addition, what is known that the signaling can work through this Rho-A kinase where it will activate finally the ROCK kinase and you will have the cytoskeletal rearrangement. Moreover, you have independent activation of MAP kinase pathway, which will involve two final MAP Ks, JNK and P38 MAP kinase. So what you notice is that in addition to the standard SMAD pathway, you are also getting signal received, and this receptor is working through multiple different pathways in which at least MAP kinase pathway, which is direct or via RAS, is coming into picture. So this is where you can see some parallels what we noticed in case of plant, where standard route of activation downstream of receptor serine threonine kinase was the MAP kinase pathway. Here it seems to be a non-canonical pathway downstream of receptor serine threonine kinase. Now let's look at some examples where receptor serine threonine kinase play very important role. One of the process which is called bone morphogenesis, it is critically regulated by a set of receptors, and this is where in previous class I discussed that how the identification of this receptor serine threonine kinase activity was through the bone morphogenesis, where extracellular extracts prepared from bone tissues were having these active ingredients which could induce the bone formation or bone reshaping and so. So we all understand that bone is a connective tissue and it has a cartilage base which upon ossification makes it hard and gives it a bone appearance. There are distinct cells which are involved in this process. Osteocytes are the basic cell type which lay the framework for bone formation and nourishment of the tissue. Then you have a set of cells which are called osteoblasts. They differentiate into the osteocytes. So you have, if I say there is a osteocyte, this comes from osteoblast. This osteoblast is able to secrete the extracellular matrix, where deposition of calcified matrix makes it a bony tissue. Then there is additional cell types which is osteoclast. These are like big mast cell which have very critical ingredient which help in the reshaping of the bone through reabsorption process, where the extracellular matrix is dissolved and the shaping of the bone can be facilitated by the activity of this. So somehow the bone morphogenesis or bone shaping or formation of bone is a coordinated activity between osteoblast and osteoclast. So normally in the BMP signaling, what happens and how it regulates the bone morphogenesis, you have BMP, which is its level in the extracellular matrix is regulated because it is generally bound to the extracellular matrix protein and it is not freely available. When it is freely available, it starts to signal, and what happens here is that the R-Smad is activated, the entire complex moves inside the nucleus and they turn on very specific bone morphogenesis program through these transcription factor molecule. We have also seen that there could be independent signal which is feeding onto this. So some osteogenic signal which are the signal for bone formation through the MAP kinase pathway, and JNK and P38 MAPK, they again converge onto these set of transcription factor which help in conversion of osteoprogenitor cell to osteoblasts. Then you have differentiation, which allows the formation of osteocytes. So what exactly will be happening here now that your bone forming signals via this canonical pathway of RSTK activates these Smad complex, which inside the nucleus is capable of transcribing the genes which will be playing important role in bone morphogenesis. So basically the conversion of progenitor cell into the osteoblast which will be depositing the matrix, then finally this will differentiate. Once the matrix is deposited and it is hardened, the differentiation of osteoblast into osteocytes will happen which will make the final bone. Then what we understood over there is that the receptor has to be here on the surface. Remember one very important thing, the availability of the BMP, bone morphogenetic protein, as a signal into the extracellular matrix. This is not available freely. This again talks about one of the things which we discussed, that how signals are regulated and you really do not have freely available signal. We will also see examples where the signal is not just not available, it is produced there, but it is in the form which does not make it active. So this PMP signal will bind, you will have phosphorylation, you will have Smad activation, and inside the nucleus you will have transcription. In previous class we discussed very specifically that this RSTK, they are heterodimer, basically dimer of heterodimers, and you will have R1 receptor type 1, receptor type 2, specific combinations are there, and there are specific combination of Smad which will serve as a R Smad in the process. So very specific transcription program will be activated depending on what signal was given. So here in case of bone morphogenesis, more so bone formation, we saw RUNX, DLX and OSX kind of gene getting activated. So this is how the new bone will be forming. At the same time, you need very specific signal to ensure that the bone is not forming extra, rather this is regulated. There is some amount of reshaping which is required so that the bone gets the desired shape. We all understand that bones come in all different forms. There are flat bones, there are rounded bones, there are long bone, there are short bone and so on. So how this regulation of bone shape happens, that is where the TGF-beta signaling plays very important role, which is important in bone shaping. We saw BMP signaling in bone formation, now TGF-beta signaling is important for bone shaping. You would not need this bone shaping event going on normally. That is why TGF-beta, which is the ligand here, is bound to the ECM, to different proteins, and it is released when there is a cue of this sort comes which says that bone shaping is required. We have seen that how bone resorption, bone formation signals were there. Here in case of bone reshaping, you will have an independent signal will be bone resorption signals. So normally what will happen, how this signaling will work, that TGF-beta will bind to the receptor following the canonical pathway. The complex of R Smad and co-Smad will travel inside the nucleus. Now they activate genes which are different types, OSX and RUNX2. There will be further regulation on this, that how OSX is activated, and to that end you have additional signal which is coming from bone resorption cues and which activates MAP kinase pathway. And here the final MAP case are very different. ERK, in previous case we saw JNK and P38 MAP kinase. Here we have ERK as a MAPK and P38 alpha beta as MAPK. Together they send the signal which is a cue for bone morphogenesis. So through this what happens, that you have osteoprogenitor cells which are generally activated and forming the osteoblasts, and these osteoblasts were differentiating. If you remember in the previous set of slides we discussed that how differentiation of osteoblast into the basic osteocytes is important. So when bone morphogenesis has to happen and the signaling of bone formation has to stop, that is where this differentiation is stopped, which serves as a signal to the osteocytes, and they activate this cell which is called osteoclast. And osteoclast is, as I mentioned little some time back, that these are a cell which have monocyte type of morphology, and they are big cells, they are multinucleated cells. It seems that they are formed by the fusion of multiple cells, and they have this edge which is called ruffled border, and a lot of granulation, degranulation event happens here. So in the area where bone reshaping has to happen, these cells orient and release the content, and this content helps in the resorption of the bone, demineralization of the bone. So the hardened extracellular matrix, calcium and other nutrients are removed from there, and that is how it is possible to reshape them back. And this activated osteoclast gives the signal to this extracellular matrix that release TGF-beta. So there is a feedback loop. The first TGF-beta is generated, which gives the signal that okay, bone reshaping has to happen, and this molecule is activated. So in some sense, when we saw example of bone formation, we saw BMP signaling to RSTK or BMP receptor in this case, which activated the osteogenic signaling, where progenitor converted into osteoblast, which converted into the osteocyte. But when it came to bone reshaping, so the molecule over there was TGF-beta, which was released as the first cue. Then through this TGF-beta receptor, osteogenic signaling of different type happened, which in that case what happened, that your progenitor were converting into the osteoblast, but this was inhibiting the osteocyte differentiation. So basically through similar set of receptor, you are maintaining a balance in the bone tissue formation, bone health and bone shape. One side you have bone morphogenetic protein, other side you have the TGF-beta like proteins. So what we have understood in this class, that receptor serine threonine kinases, not only they have a canonical signaling pathway, which is very different from what receptor serine threonine kinase do in plants, but they have multiple non-canonical modes as well, and one of the non-canonical mode is the MAP kinase pathway, which bears the similarity with activation of RSTK in plants, and that is where this may involve this MAP kinase pathway. In addition, it will also activate growth through RAS mediated MAP kinase pathway. Importantly, the interplay of two molecules which respond through receptor serine threonine kinase, the two molecules which are TGF-beta and BMP, you maintain the bone health homeostasis, and bone formation and bone resorptions are having an equilibrium, so that the bone is shaped fine, bone is growing fine, and its health is maintained. Thank you.